Okay, good morning and welcome back to the lesson. We were trying to discuss about how to differentiate gametes from embryonic stem cells or other cell types. And we also have discussed in the last class about why we are supposed to or try to have a motivation for the reasons why we should be doing these things. So there are two problems. One of it is there are genetic defects. The other problem is that the dwindling fertility rates in the humans. So we also, to a quick recap is X and Y. Y chromosome is what differentiates something from female to male, in the sense that our default is female, right? Our default program is female, and a variation to that is the male. And the reason, or at least partly, or central to it, is one of the genes that is referred to as SRY. Throughout the lesson, also, we will discuss a few of these factors, but we should understand that they are much more complicated or complex, and there are many other factors which we cannot discuss at this time. So, if there is no SRY, then the a gonad will develop a structure in general which is indifferent, or you can also call it as bipotential. And if there is no SRY, then it grows or results in the synthesis or the development of ovaries, and then in which you'll also have the eggs. Whereas if SRY is present, then testes are developed and other male organs, and then it grows, it will produce sperm. Okay. So we have seen the gonads in general, and what we are trying to learn is if we want to derive from embryonic stem cells, we want to derive, or any of these cells, we want to derive gametes, then we should have understood what it makes or how the development of gametes happen, right? That is what we are trying to do. So around or before the early trimester or so, there are some gonads formed, which you call it as bipotential, meaning that until then it is kind of unisex, in the sense that it can either form the ovaries or the testes. So they are called as bipotential. And there are ducts also formed, Mullerian duct and Wolffian duct. And if there are no male hormones, then the Mullerian duct will develop into oviduct, and the bipotential gonad will become ovaries. And then we'll have the development of cervix, vagina, and other things. So male reproductive organs. Whereas if SRY and others are present, there are male hormones produced eventually, because of which the Mullerian duct recedes or regresses, and the Wolffian duct is developed, which will form the epididymis and vas deferens, and of course the testis. So in a way, when you don't have the male hormones, then it is the female, right? And otherwise, you can also see some people who may have dysregulation because of any of the male hormones are missing, then they tend to develop female characteristics. I mean, sexual, the organs or developmentally. So let's just have a look at a few more things. Uh, during the embryo stage, it forms a genital ridge, which is going to be the future of the gonads. And because of these factors, we don't have to memorize all of them, but it will be good idea if we just at least know that there are complex or multiple factors. And genital ridge to bipotential gonad, this is common in both males and females. But in males, when there is SRY, and also there will be an expression of SOX9, and then the bipotential gonad will now differentiate or develop into structurally testes. And they will have Sertoli cells and Leydig cells, which will eventually have the anti-Mullerian hormone, the production of anti-Mullerian hormone and testosterone. The testosterone will activate the Wolffian duct, or will allow the Wolffian duct to grow into the male internal genitalia, like the epididymis, vas deferens, and so on. And the anti-Mullerian hormone is actually going to make the Mullerian duct or regress, which in the sense it will go extinct or degenerate within the males. So they are not there. Until here, say in this case, you have both the Mullerian duct and the Wolffian duct. And in females, the Mullerian duct will develop, and in males, this one will develop, and the other ones will get degenerated. So when there is no SRY, then there are other factors, DAX1 and WNT4A, 4A, and so on, will help or will result in the differentiation or the development of an ovary, and which will develop the follicles, and follicles will produce the female hormone estrogen. And then they will eventually, the Mullerian duct will develop into the female internal genitalia, including uterus and cervix and other things. So we are not actually concerned with what is going to happen with the human body itself or the parts. We want, as a part of this, we are trying to develop the gametes. So I'll skip this. We can have many number of papers that we can read, and there will be some alterations, but you will have so many of these articles trying to describe these things. So here is what we should actually focus on. What we are trying to do is to learn about how primordial germ cells are forming. That is the first step, right? Because from zygote, or I will say it as ICM, the germplasm is from the primordial germ cells. The somatoplasm is something like primordial germ layers, and the germ plasm, okay, ICM will give rise to PGC, and that will give rise to gametes. So if we want to learn about or try to differentiate them as an application to achieve gametes, then we should try to understand the biology or a little bit about it, some introduction. So here is a growing embryo around the embryo stage 5 or 6 and so on is written as that. And then the proximal epiblast, within the proximal epiblast, somewhere around here, these are the cells that are being described here. So these are some cells of proximal epiblast. And BMP is the BMP, these cells develop competence for BMP4, and then they become little bit differentiated, and then one can find the expression of fragilis, and that is where what was proximal epiblast is now differentiated because of the influence of BMP4 and so on. And then with time, the pre-GC precursors, they are going to form. That means these competent cells. Of all these cells, some cells will produce BLIMP1. Then they are differentiating into some of the cells. Those cells that are competent, and now they become PGC precursors with the expression of BLIMP1, and then they form what we now call them as primordial germ cells. They have expression of fragilis, they have BLIMP1, that is where they have become the PGC precursors, and they also have an expression of Stella. I hope you also remember, you will remember the word Stella, right? In one of the chapters where we were learning about the global demethylation of the genomes and so on. And we learned about what factor, if we delete it, also results in the demethylation of the imprinted genes. There we came across Stella. If we deleted Stella, imprinted genes also may get demethylated. That was one of these papers studied some time ago. Anyway, I hope you understood the series. The proximal epiblast becomes competent by the expression of fragilis. Out of them, some of the cells will express BLIM4, and now they are primordial germ cell precursors, which then those that have BLIMP1 expression will have the expression of Stella, and then now they have formed the primordial germ cells. So from here, it has formed an embryo. Assume that from embryo we have the primordial germ cells. So yeah, I forgot to mention, yes. So fragilis is important for cell-cell adhesions. And then BLIMP1, what is it doing here is suppression of somatic cell development. That means it is kind of determining these cells towards becoming the germplasm, right? So these genes which determine the somatic cell types are all repressed. And then Stella is involved in the regulation of chromatin, like we also discussed. That is another way, or it came up in the hits in there about the imprinting status. Okay, so if I am trying to check, if I'm trying to determine some blasts, epiblast cells, and then I want to know that they have primordial germ cells have formed, and from primordial germ cells, I want to know that gametes have formed. There are a series of differentiations. Okay, it is much more complex than what we are discussing, but this is sufficient for our understanding. So the first one, until here, we have seen in the previous one, where we know that there is expression of BLIMP1, fragilis, BLIMP1, and also Stella, right? And there is also expression of OCT4. As I said, different studies will have focus, and different people will keep supplying more information. That's how science grows. So the inducers are BMP4, 2, and 8B, and they will cause the PGC speciation. And if I didn't mention it, the primordial germ cells form somewhere in the outside of the extraembryonic membranes tissues. I will try to discuss this at a later time, or maybe let's see, if this is a growing embryo, this is where the gonadal ridge is, and we have somewhere around this place the primordial germ cells form, and from there primordial germ cells should travel and then colonize the genital ridge. This is the genital ridge. Okay, simplistically, that's how you can see it. So they form somewhere outside, and then they travel in, and then they reach the genital ridge, and based on further expression, they become either male or female. They will influence the sexual characteristics of the embryo in terms of developing the internal genitalia or external genitalia and so on, and also the type of gametes formed, either sperm or egg. So primordial germ cell speciation, and after that they will start migration around this stage. When they are moving, that is when you will also have the expression of CK, Stella, and so on. And there is a gradient of soluble proteins from the genital ridge. How would the cells move is, say, we are unsure completely, but this genital ridge is producing some signals. Okay, so if it is constantly producing, you will have higher concentration in the genital ridge, and lower concentration as we keep moving farther away, isn't it? Say if this is the genital ridge and this is continuously producing more and more of the signals, you may have some 100 molecules here, some 50 molecules, 25, say 12, and so on. So the primordial germ cells will keep on moving towards the gradient, increasing gradient. And that is how they probably reach the genital ridge. There are several defects where sometimes the primordial germ cells may take a wrong route. And if they reach some other body parts, they usually may undergo cell death, apoptosis. But there is also a possibility that they might sometimes develop into tumors. I think if you have read the news enough, you will find out. I want you to find out a sports personality who had this disease. Anybody? Or you can try to find out and let me know. It has something to do with the localization of primordial germ cells. Okay, so the migration happens, and then once they reach the genital ridge, they will colonize. That means they will undergo mitosis, several rounds of mitosis, so that the whole genital ridge is filled up with these gamete precursors. You can talk of them as. And during that time, they will have expression of these molecular markers. They are increased these levels within the cells, and they are now colonize the ridge, and they will undergo arrest. In case of males, they will be, in case of, excuse me, in case of males, they will be in G0 phase. So right when the embryo has formed, they reached into the genital ridge, and within the genital ridge, they will just undergo, they will enter into G0 phase and remain dormant until the next 12 or 14 years. That's in males. In humans. And only upon maturity, they would re-enter, and they would cell cycle and undergo meiosis and keep on producing mature gametes. So we'll just look at what would happen to the male first. So once they have reached, they colonize the whole testis, and then they would go into G0 phase, 10, 12 years of sleeping, then puberty, and then they would start producing mature gametes. And this is TEKT1. This is what is the molecular marker in mature or sperm. Whereas in females, they would, after colonizing the gonadal ridge, they would enter into meiosis, and there they would stop in meiosis. So they are arrested. In the males, the arrest is at G0 phase, I mean, prior to meiosis, whereas in females, the eggs are arrested in between meiosis. And this is GDF9 is one of the molecular markers that which is specific for eggs. So what we are trying to see here is what induces these processes and what event is occurring if it is in the normal body, and what are the molecular keys within the cell that is kind of consequence and also for, requirement for the next step. Okay. So we'll use these as molecular markers. That means if there is any cell that is producing TEKT1, that means it must be a sperm. Or if it is producing GDF9 in abundance, then it is egg, like that. Or if we know that some cells are producing these three, that means those cells are at the stage similar to that of the primordial germ cells after or prior to entering meiosis. And for meiosis, we have these proteins that can be used as markers. So this is the same thing, shown differently, of course, but you can try to go there. So fragilis, BLIMP1, this is where we are having, this is in the epiblast. Then we have blimp and stella, OCT4. Then migrating cells, we have c-kit. And post-migration, you will have DAZO. And post-migration, post-colonization. That means here they have just reached, whereas here they have colonized. Then they would have also expression of VASA. If there it is, if they're undergoing meiosis, then it is CMOS. And if it is sperm, you have TEKT1. And if it is egg, then it is GDF9. Okay, these are markers. Markers means here, if a cell has c-kit, high expression, then it would mean that these are primordial germ cell-like, and they are at the migration stage. Like that, we use these as markers. These are protein factors that can be detected by several techniques. So all differentiation happens by two factors. One of it is from the signals that would come from the niche, and the second one is what genes are already expressed, that is the intrinsic regulation. So normally in a cell, as we discussed earlier too, there are two properties. One of it is self-renewal, and the other one is ability to differentiate, that is the plasticity. And here the first one is self-renewal, or you can call it as also stemness. That means the division is asymmetric, right? So how that happens is maybe the half of the cell is experiencing extrinsic signals from the niche. And that niche, so some of the proteins could be localized onto one side. That means the future cell that is going to become this, has different experience or signals compared to the new cell that is forming. And that is how this cell can be, or the new daughter cells, one can remain as a stem cell while the other one can become differentiated. Of course, as I was telling you, there is also about gene regulation intrinsically, what genes are expressed and where they are localized. So this is still one cell, right, at this stage. And how can it be that they can become two different? It will depend on what type of proteins are localizing to the different poles. So upon cell division, the inside one of it could be a stem cell, and the other one could be a differentiated cell. We have learned about several differentiation strategies in the last lesson. We know we have heard about spontaneous differentiation, where something like skin and epithelial type cells can come up, and by addition of specific growth factors. We have also learned about co-culture and genetic manipulation. Excuse me. Yeah, sorry. So where was I? Yes. When we learned about these genetic reprogramming, epigenetic programming ones, what we are trying to do right now is also the same thing. We learned about the biology of how to the gametes, and we want to differentiate them by using artificial mechanisms. In that, we learned about nuclear transfer, cell fusion, cell extract, and cell explantation, right? There are advantages and disadvantages of each of them. And of course, there is also genetic manipulation if it is there. So we are going to see several others, which several papers, a few of them at least in detail. And they might have used any of these strategies. I'm not going to really discuss the strategies that they have used. What I am interested in, what they have found, because the strategies we already discussed in great detail in the last class. So the first paper is that we are going to discuss is about Toyoka et al. From 2003, where they first produced sperm-like cells from mouse. So some experiments were done in mouse, and some we will see in that are from humans. So they have used transfected transfected mouse embryonic stem cells. 1 second, please. They transfected mouse with post-migratory cell marker. When you say transfected, what do you understand? It is a genetic method. So they would have taken cells, and the cells that are taken are embryonic stem cells, and then they would have overexpressed by putting, they might have made a construct with MVH, mouse Vasa homolog, and then they would have put it into a vector, and then they would have transfected into embryonic stem cells. So because there is a strong promoter, you will have the mouse Vasa homolog expression, and because of which the embryonic stem cell has become something like a sperm or a precursor of sperm. Okay, so the approach is a genetic approach because they're using genes here. And in the culture conditions, there is also they have added kind of, you can think of it like this. The MVH here, they're using it as an intrinsic factor, right? As I was describing, there are two things. One is you need an intrinsic factor, and you also might need extrinsic factor. And the extrinsic factors, for the extrinsic factors, they have used, those are the culture conditions to stimulate differentiation. Is they have removed LIF, leukemia inhibitory factor, allowing the formation of embryoid bodies. EBs are embryoid bodies. What are they? Embryoid bodies are something grown in the culture, in the medium, which resemble that of a fetus, right, or an early embryo, and that has primordial germ layers forming and so on. And they have done something else as well. After that, they have added BMP4. I hope you remember BMP4 is here. Yes. So that is important for the stimulation or the specification of primordial germ cells. That means whatever cells were responding, they would start to become precursors of primordial germ cells. And then they have, after detecting the MVH positive cells, so they have taken the cells that are having high expression of MVH, and then they transplanted into host testis. That means they have taken a mouse dissection, and they have taken the into the testis of the mouse. They have put these cells, MVH. And what they found was the mouse produced sperm, which is the one that have, which is the transplanted cells. Okay, so what are they proving is important. As I said before, probably there are two things. One, people do experiments to prove something as a proof of principle, meaning that it works. We can get ideas that maybe we can use embryonic stem cells to produce sperm. And then we can also, later what will happen is we should have developed a model why it should work. And the third step is proof of principle. That means you have to show that the key step of it works. And then from there on, optimization will work. We will keep on optimizing until what we get, and then we we usually go through them as kind of therapy or for clinical trials and so on. So I hope you understood the essence of this paper. So let me repeat. This is the first time they have produced sperm-like cells from embryonic stem cells. They used the genetic method by overexpression of MV, which is like a VASA homologue. The VASA is here. And then they have used as in the culture medium, they have removed LIF to allow the formation of embryoid bodies. And then they have cultured those in BMP4 to stimulate the formation of primordial germ cells. And then once they are formed, they have put it into mouse testis transplantation. And then they have found that the sperm is produced. The question would be something like, how would we know if the mouse has testis, which means that it has its own spermatogonia and other things? How would you know the ones that are produced are from not the earlier ones, but from the newly implanted ones? So usually what may happen is that we might choose a genetic factor, genetic element or so, that is special to these cells that we have taken here. Okay, and based on that you can do. And second point is, when they are transplanting, usually they will try to clean up a little bit of the host testis so that the earlier spermatogonia are removed. Of course, complete removal is impossible. That's why we will need to go for molecular characterization of the produced sperm. Say, for example, we have taken embryonic stem cells which have gene X, okay, or maybe GFP. GFP is green fluorescent protein. We have constructed it, and then we have put it into testis. And if there is any sperm that is producing GFP, that means it must have come from the transplanted cells, right? The original host may not have, or we did not manipulate it to have GFP. Those are marker genes. Okay. So the second one is in the previous one, we went from embryonic stem cells to sperm-like cells, right? That participated uh, in in spermatogenesis. I mean, meiosis happened, everything, but we don't know anything about their potential for fertilization, because sperm is special in the sense it you need to have the apparatus for the fertilization to occur. So in a different paper, Jensen, they differentiated mouse embryonic stem cells into mature sperms. They added retinoic acid to the culture media, and then they identified markers for the male characteristics, and also they have studied their methylation status, like we have discussed in the previous classes. And they demonstrated that the male germ cell development and meiotic maturation are possible from embryoid bodies. So what they have done is, say, we have sperm has been produced, and whether it can form a zygote or not is one question. If it cannot have its own material, meaning that own complete differentiation, that is, a mean with acrosomes, tail to stem, and so on, how does it, can it still, is there a way to allow the formation of zygote? So for this, they have done something similar to that of ICSI, intracytoplasmic sperm injection, and they have shown that these zygotes the sperm that is produced, okay, mature sperm-like, can form a zygote and can go to blastocyst as well. But then how good is it, whether the methylation status, epigenetic imprinting, and all those things? I hope you remember that. The germplasm or the gametes have the epigenetic wise, it has special role in that. And one has to be very careful. If there are alterations, there could be problems with the way the embryo develops. Until blastocyst, at least, they did not have that much of a problem. But further studies are required. So in one paper, not everybody goes completely. Science walks in this way, right? And that is when it is most fruitful. It doesn't walk this way. It's not easy. So different labs working may work till here, others may take it a little further, and others may take it a little further, and so on. That is how we try to, the science is self-corrective. Typically, where self-corrective means science will find its own mistakes and try to, if there is a mistake here, the study here could actually try to correct that mistake. And that is how it works. It doesn't work this way because there is always a possibility of human error or some form of error. And they don't work this way. It usually works in this way. And that is what we have seen. In the previous one, somebody has done from embryonic stem cells to sperm-like cells. And somebody now has done almost similar and with a different strategy, has taken it to mature sperm. And somebody will go, these people went beyond to show that it is mature by allowing the formation of at least the blastocyst stage. But yeah, whether it will give into a functional and normal person or not, or offspring, is a difficult question to answer at this stage. But I think the results that we are discussing are the earliest ones. Now too, it is still in the same stage in a way. There is progress made, but it's not that nobody has tried to produce complete organism. I think there are some examples with mouse, none with humans. I think there will be problems with ethical committees agreeing for such kind of research, especially when it is involves human embryos. There are problems to go ahead. So here is something about, somebody has, in the previous ones we have discussed about coming until from embryonic stem cells to sperm-like gametes to produce the blastocyst. Here is a research that is to show that it can produce a complete organism with a mouse. So they have established cell lines from mouse embryonic stem cells, and then transfecting two marker genes. Again, transfection, when you say, you are putting a gene like this. They have used two proteins. One of it is GFP, green fluorescent protein, and red fluorescent protein, linked to promoters of STRA8 and PRM1. So what they would do is, this is promoter for STRA8. And the other one is promoter for, I'll put P is for promoter, PRM1. STRA8 and PRM1 are expressed, are markers for development of into sperm. Okay. So they have put GFP, and here they have put RFP. That means if there is a cell in which from its genome there is expression of STRA8, what does that mean? It means that the promoter of STRA8 is active. If I have the same one, say for example, if I have the same promoter, okay, I have the same promoter as is present for STRA8 here, except that I have GFP here. What would we expect? We will also expect the expression of GFP. So all the cells in which STRA8 is expressed, you'll also find the expression of GFP. Or look at the other way. If GFP is expressed, green fluorescent protein, you can see that, say you have a culture medium, and on the slide you are able to see that some cells are giving fluorescence and some cells are not. What would that mean is in this particular cell, we have the expression of STRA8 from its genome. That is the indicator. So if STRA8, if a cell is showing the expression of GFP and RFP, which means the cell must be expressing, or the promoters of STRA8 and the promoters of PRM1 are active, which means they are undergoing gametogenesis. Okay, it's a genetic strategy to identify which of these cells are differentiating into gametes. So similar strategy, retinoic acid was added to embryoid bodies. Embryoid bodies are formed, and then some cells were found with motility, and they are coming out of, say, this is embryoid body. Some cells are coming out which have motility, and they are expressing all the meiotic and post-meiotic markers, which means they have completed meiosis or they are undergoing meiosis. And they collected those cells and put it into testes of sterilized mice, and those gave rise to haploid sperm. But they are not highly motile, they are little bit motile, not highly motile as that of a normal one. They have taken the sperm, and they, through ICSI, they have been able to develop the zygote. And once they reach the blastocyst stage, they would have put the embryo into a surrogate mother. And what they have seen is that at the end, they were able to get live offsprings. One of the problems is that most of the pups died prematurely. And they have seen that some of them had overgrowth, some of them had growth retardation, and there are very short lifespans. The reason for it is probably the abnormal methylation patterns, where the failure to establish proper germline imprinting. If you remember how much the imprinting we have discussed in the previous class, it is the primordial germ cells, there is something to happen, erasure, mostly as a general model. And gametes, by the time they come to the gametes, they should have established. For these events to occur, and to ensure that these events to occur in the normal embryo, the cell will receive all the signals and other things required for these events to happen. And then gene expression will determine what is going to happen also. In the protocols that we are discussing here, we cannot absolutely mimic at this stage of technology and knowledge. We cannot mimic the processes, the biochemical factors, physical interactions of the cells in the test tube. We cannot do that at this time. And so, which would mean that the imprinting status, the erasure and establishment of the imprints is not happening as per our desire. And therefore, the offspring they have formed, that is a huge step forward, but it is not a perfect step. There are so many things that we need to relook and try to have imprinting in a better state. So before I will just take one more point here and the next slide, and I will stop. I want you to look at this table, and now I want you to also think about this. XX, can it become a male or a female? It will become a female. XY, can it become a male? You'll also see a pattern here. XY can become a female-like characteristics, okay? But you would not find any XX, a female, giving rise to male. What I mean by that is, say here, we might take XX, the sex of the cells, mouse embryonic stem cells. Female, we can take from a female or a male. And through these techniques, we can generate oocyte-like cells. Whether offspring is formed or not is no or not determined, and so on. Okay. Here is XY. It can either become a sperm or a male, or a oocyte. In vitro, it becomes, they have, this is a summary of a table summarizing many papers together. So it forms sperm. And I want you to find if there is any female. This is something that will not happen here. That means a female cell giving rise to a oocyte or a sperm, sorry. Right? So that is one thing. That is how you can also try to look at. XX is sperm. XY can form oocyte-like cells. Oocyte. If it is a sperm here, then it's always XY. Okay. Spermatids XY. And say XX and XY, they can form oocyte-like cells. It's just that to mean you need SRY to become something like the sperm or eventually the male type of gametes. Okay, I just wanted to bring that to your attention. When you see a table or a graph, you should try to build a pattern out of it. And when I looked at it, my pattern is, because I was looking at other parts, reading also, I had the perception that female is the default program and male is a deviation from the norm. That means there should be something else to become the male. And that at this stage, we can consider SRY as an important factor. Whereas in a male, how can it give rise to oocyte-like cells? If for some reason SRY is not expressed, then it is as good as a female, isn't it? If there is a Y chromosome and Y chromosome SRY minus, then it is almost as good as a female, right? That I think you will be able to think on that a little more. So this is the last slide, and then you will have to read this chapter and get back. And this one is to highlight the human embryonic stem cells. This is one example that I have taken. The previous ones, all of them are from mice. So they did almost the similar ways. Vasa expression following the appearance of germ cell precursors. They have taken embryonic stem cells, and then they have grown it, embryonic stem cells, and then they've kept tracking these markers. And then those cells they were taken and then tried to analyze what is happening. There were genes involved in meiosis, but meiosis did not complete. Okay. In mouse models, it is a different thing. So what is true for mouse is not necessarily true for humans. There could be a lot of similarities, but one should also expect that there are deviations from it. Okay. So the problems, as I was describing, in vivo, the settings are different. There is germ cell line lineage, primordial germ cells, and differentiation. There is the methylation patterns and many things that happen during this process in vivo. Whereas in vitro, we don't have this, and that is what is going to compromise the health of the offspring. Okay, so we need to still study a lot more about how to properly allow the somatic imprinting, to happen. Whether the methylation is going to be erased completely as it is in the normal in vivo ones, and how the imprinting patterns can be made during gametogenesis. These are some things that we still have a long way to go with. So this is the last slide. And from here, this is an assignment that you need to work. The central dogma is it is thought that every female is born with a limited number of eggs or ova. And each week, each menstrual cycle, one egg is released. And say if the female started at the age of 12 and about age of 45 or so, ovulation starts at this age, and for these many years it would keep on producing one egg at a time every menstrual cycle, a month, assume that. And about 45 years or so, that is menopause and stop of ovulation. Why should it stop here? Why not give until lifelong? Because if you look at the male, for spermatogenesis occurs from about 14 or so to until the person is alive, almost till 90 years also. So what they think is the central dogma in this case is that females are born with limited number of eggs. Once they are exhausted, there is no more production of oocytes. That is what they think. Okay. Some people challenge this. Some people agree with this central dogma. Some people don't agree with this. So I want you to read the paper in the material that is given to you in this chapter, where differentiating gametes from embryonic stem cells. There is a paper description on a paper, Johnson et al. Then I want you to read what is that they proposed. I mean, this is the paper in the material that we have. And there is a nice discussion which I want you to discuss, where they have discussed about 3 different papers. I want you to read the experiments and have your own justified comments with regard to the central dogma. The question is, are females born with limited number of eggs? Or you can also have, is there a possibility for eggs to be produced after birth? Eggs to be produced means new oocytes. Can they be made anew or not? Okay. Yes, I'll stop here. If you have any questions, I think I missed. If you have any questions, we'll discuss in the next class. Okay. Thank you. Any questions? I forgot to ask. Please read this part. Okay. It's important that you also read the material. So I am trying to facilitate your reading through when I discuss, you can read through the paper well after the class. That's one of the objectives, of course. Please do that. Okay.
Can you share these articles?
Yes.